Thank you everyone for being here. Again, as Ben said, my name is Ellen, for those of you who are just joining us now, and I am joined with Aaron Schirmer, who is a professor at Northeastern Illinois University. So today we're going to talk about all the work that we've been doing at KBase over the summer, because we realized that there is a need to help educators teach bioinformatics online, especially as we are transitioning from doing more in the classroom work to virtual learning and teaching. And so for those of you who are new to KBase, KBase is the Department of Energy Systems Biology Knowledge Base, and it is a free and openly accessible data science platform. So this platform will integrates many different resources for bioinformatics analyses. And this last summer, we launched our community of educators who we've been working with to deliver these new resources. And so this organization and community was implemented to create different resources for educators so that they can provide their students with hands-on data science research and analyses experiences. And so the reason this came about was because we realized that the narrative is a unique resource. It is essentially built on top of a reproducible Jupyter notebook instance. And so this Docker image is shareable so that someone can see exactly what research you are doing, from data to results, including any analysis, as well as different notes and conclusions that you come to. And so our community of educators spans from high school teachers to college professors, as who are teaching, undergrads, graduate students, even we have some postdocs who are a part of our community, and undergraduate teaching assistants who have also helped put together these different narratives, which are our reproducible notebooks that KBase uses. And so the different resources that we have created I'm so sorry, I missed the slides. So the different resources that we've created are a concept workflow, the KBase Educators Org, which includes narratives, and then we've also gone through support from using best practices, as well as creating a community and different aspects that that community can use. And so with that, I am going to share what these different pages of our support look like. So when we go back to the concept workflow, what we've created is essentially some different concepts that we've built on, from genomics, metagenomics, phylogenomics, and pangenomes. And so each of these different topics uses a narrative that is built as a module. So these different modules are standalone, but work together. So they're all embedded together. For instance, with genomics, you go from one narrative that is based on assembly to annotation, and then you have different options to do more exploration, like analysis, comparison, and these flow together across the different topics. And they all kind of accumulate around pangenomes. And of course, these are also buildable to do more topics and more ideas. And this is one example of a static narrative. So this is not changing, but it does live as a editable narrative as well within KBase. And so here we have our different topics listed out, and these are all connected to the different narrative modules. We also have different student resources, and this is a version of the students. So within the educators version, you'll actually see a link to the org, and as well as the drive that has some additional peer shared resources for others to use. And so from there, we also have our KBase Educators Org, where you can find this and other items. So this is what the about page of the org looks like. There is information on how to join the org. So if you come to this page, which is narrative.kbase.us/hashtag/orgs/kbase-educators, you'll come to the page that will look more like this, and there'll be a request to join. So you can request to join. Please send us an email at engage@kbase.us and let us know what course you are teaching, the topic and the level, when you will be teaching the course, and how we can help you out. As well, if you are interested in working with our educators to co-develop these similar resources to what Erin's going to walk through as an example, please let us know as well, and I can connect you. Within the KBase Educators Org, you'll also see a bunch of narratives that we've been working on. And we have both educator and student versions. So the student ones are going to look more like blank workbooks, while the educator ones will have all the data and everything worked through. And so in addition to that, we also have different items of support that we have put together and created. And so one of those is a best practices. So along with the community putting together these narratives and these module workflows, we've also done some troubleshooting and figured out some of the better ways on how to deliver KBase and use it in the classroom. So from more teacher support to how to encourage your students, how to do different things like create your own organization for your classroom, to sharing a narrative, as well as links to all of our different resources, figuring out the different teaching logistics. We have it all in this document, which is of course growing and changing. So as we learn more, we're able to update this and share more with you. So we also have, in addition to this, we've been working with educators such as Erin, as well as another group. And really this community of educators has kind of brought us from nothing to everything we have today. And so really this came about through conversations about discussing the need and really what topics instructors are teaching this fall and this coming spring. And that's how our topics came about. So we are looking into expanding into metabolics and transcriptomics and focusing on these concepts in the future. So if you're interested in those, please shoot us an email at Engage, and we can connect you with some different educators. I see Carlos is on, and he's one of our part of our community, and he's very excited. And with that, I'm going to pass it off to Erin so he can talk about his experience.
Great, thank you so much, Ellen, and thank you all for coming today. So I came to KBase sort of randomly. I received an email about a workshop that they were doing in the summer of 2019 at the University of Chicago that was just sort of on the KBase platform. I went to the 2 or 3 day workshop, learned about KBase, and as they were sort of laying out these different tools, I realized what an amazing tool this could be in the classroom. And I talked to some of the organizers, and they mentioned, hey, we're trying to put together this community of people. And so this past summer, we met multiple times, and we talked about how we might take the tools that are available in KBase and move them into the classroom. And for me, as was mentioned before, I teach at Northeastern Illinois University. This is a primarily undergraduate institution. We are a minority serving institution here located in the Midwest. And I teach across our curriculum. I teach everything from introduction to biology for non-majors up to courses in our master's program in sort of my area of specialization. But the course that I want to talk with you all about today is my genomics and proteomics course, which is a lecture-only course. This is sort of an elective in our curriculum. It has a prerequisite of general genetics. So you have to sort of complete our intro or our general biology series, and then you have to take general genetics, and then you can get into genomics and proteomics. So this is for upper division undergraduates. And we've also made this course available to our graduate students as well, realizing that this was a topic that many of our master's students might be interested in. So the course that I was sort of thinking about for KBase and designing this module for was really for a mix of upper division undergraduates and graduate students. And at this point in their biology careers, these students really have very little experience or interaction with any of the omic disciplines. And so this course is really an introduction to the fields of genomics and proteomics. And we use a combination. The course is lecture only, but through lectures, discussions, and hands-on activities, I try to expose the students to, again, the basics and in introducing them to these fields. And Ellen, would you mind going to the next slide? Thank you. And so how I incorporated KBase. So I have the lectures that I'm going throughout the semester. And then normally in class, I would also go into little breakout rooms. We would have students, spend 20 minutes here, 30 minutes there, working on different hands-on. And in the past, this has been going into some of the NCBI databases, doing blast searches, doing some simple annotations, things like that. Now, this past semester was unique because we were in a remote environment. And so I infused KBase into the course using 3 homework assignments. And each one of these homework assignments contained 2 to 3 of the KBase modules or narratives that Ellen was talking to you about before. And sort of infused into these different modules are a series of questions. Some of them are pretty straightforward. Are you looking at the right thing? Are you interpreting this graph? And then some of them are more, are you understanding what kinds of analyses you just did? And then some of them are more thought questions. If we change this, what would we expect to happen? And so each of these assignments contained 2 to 3 modules, and there were a total of between 20 to 40 questions that the students answered using the KBase tools. And I gave the students 2 weeks to complete each one of these assignments. And if it's possible, I will try to share my screen. And take you through. Can everybody see the narrative?
Yep.
Okay. So, so this was actually a narrative that was one of the Stephen Biller and Ellen developed this narrative this summer, and this was the first narrative that I used for the students, and this is sort of on a genome assembly. And so up front in all the narratives, there's some, who is this narrative geared for? What are the learning goals, the biological topics and concepts, and then a general description. And then it sort of takes you through some of the background, an overview of what the students are going to be doing in the module. There are often links out to different papers and different detailed reviews. And then you can see that then we get into the different apps that the students are going to be using to import data, analyze the data. This one is the student version. So this one is not completed. So this is one where the students would go in, they would import their data, they would run the narratives themselves and analyze the data. You can also see here that we have questions that are infused throughout that the students have to answer on the material that they're working through. So there are 2 versions of the app in the Educator org. One is the student version where they can work through all the apps themselves, and another is the teacher version where all the apps have been run, all the answers are there that you can then work through and figure it out. And so again, you can see these questions are highlighted. These are just sort of, are you following along? And then I believe if I scroll down, there are also comprehension questions that are sort of a little more detailed, take a little more thought, and a sort of more critical analyses that the students can work through inside of this module. So that just kind of gives you a very quick overview of what that module looked like. And then in my next slide, so along with these modules, so the students work through these 3 homework assignments. They work through this series of modules. Sorry, Ellen, can we go back one slide, please? So they work through these 2 weeks. I also, as part of each of their exams, there was a practical portion of the exam where they used the skills and apps from each of these homework assignments as part of their exam. So the module that I just showed you was getting students working with staff and looking at antibiotic resistance and analyzing their genomes, comparing antibiotic resistant and non-antibiotic resistant staph. And so, for example, in one of my take-homes that follow, or one of my exams that followed that, the students applied the skills that they learned to a novel data set. And so they had to use similar tools, use similar apps and analyses to look at cholera, and then try to interpret those data and figure out what, if they thought cholera would be resistant to this particular class of antibiotics or not. And so that was really successful. So they were using the KBase sort of through these homework assignments, and then they sort of showed me that they understood those skills and how to use the apps as part of their exams. And so the 3 homework assignments, which is on the next slide, the 3 assignments that I gave the students was, we started with a sort of a genome analysis, which took the students through assembly, annotation, blast searching, visualization of the genomes in staph using circle plots, and a little bit of metabolic modeling. I also did a comparative genomics module where they used genomes to develop species trees and do proteome comparisons. And then the last module that they did was sort of building off of pangenomes and taking them through how to build, visualize, and compare those pangenomes. So this was one approach that I used. This was building off of modules that I developed, as well as modules that others developed in the summer across sort of our summer working group. And so this is just one example of how this can be used. But as Ellen mentioned before, we had high school teachers and we had people who are and faculty who are dealing with upper level PhDs and postdocs. So really there's sort of a tool, there's a narrative for everybody in there, and each one of these can be customized, I think, to fit your specific goals. So again, this is just one example of how I use this in an upper level undergraduate course, but there are many different permutations that can be used. And I think with that, I will pass it back to Ellen, and I know we'll have a little bit of time at the end, and I'm happy to answer any specific questions that folks have.
Thank you, Aaron. Yeah, and so as Aaron said, there's actually some advanced, more advanced tools if you were interested. This last summer, KBase was a part of the summer school that PNNL and EMSL, so Pacific Northwest National Lab, and one of their user research facilities. So they put on a summer school, and this summer it focused on microbial dynamics modeling across different scales. So from the very small as well as up to the scale of an environment. And now KBase actually has a page where you can check out this course. It includes on this page There is everything from lecture videos to the presentations. There are also the example narratives and the supplemental like readings. So if you're looking for some more inspiration, this is a great place to go. This was developed for early career scientists. So might be a bit advanced for students who are just learning how to use KBase. But this is another resource that is there. And there is a similar organization, which is the 2020 Microbial Dynamics Summer School Organization in KBase. So just like the KBase Educators Organization, there is this organization that has all the different narratives that they used. And if you are interested in developing your own narratives along with all of the narratives that are available within the KBase org, you can also check out some other resources. I have created this module skeleton for education that is this is the structure that we used for our education modules that we created this summer. But really it just kind of goes through as an example for how this looks. There's even a markdown guide. These markdown cells, if you copy this to yourself, so you can actually, like I've shared this, so you can share with your class, you can save it, and then if I wasn't sharing it, it would look like this, so I can make a copy of this narrative. And that's how students would typically do a copy. But if you look at the markdown when you're in the edit mode, you can actually double click there and it looks a lot different. And now you have all the different code that shows how you can, how you highlight things or change color or add images in to give that supporting structure and information to your students, similar to a textbook or any other sort of workbook. And really how we've built these out are kind of, being able to give students the context, the background within the platform itself, so they don't have to bounce around as much. And then also, being able to create these different links. So I can jump from, how samples were processed outside of the platform through within the platform, looking at the different data inputs, data exploration tools, results and analysis. I can jump down to reflection using hyperlinks, and then even set up next steps. So for all of these narratives, they are all linked together, which is really great. So, if you're in one narrative, you can just jump to the next one after you finish one of the narratives, or which we've built out as different modules. And so that is just one of the resources that we have within the Q&A. I've linked that narrative. I've also linked the statics and version of the genomics, which Aaron showed, and we have our best practices documentation, as well as the concept workflow that is available, that is here with all the different topics that we have covered. And so, again, I just want to reiterate joining the community. So along with these different resources, so the tools that we've already built out with these different narratives, to the different narratives that have either resources that link out of KBase or show you how to create your own narrative for your students, creating your organization for your classroom, and really walking through and providing the support. We also have a KBase user Slack and an education channel. Within our best practices, we also have a guide for Slack that was created by another organization. And so this is just like a really quick run through of how to use it if you're not familiar. But we have a specific channel for educators, so you can actually have conversations with other instructors. Also talk to the KBase staff if you need immediate help versus submitting a JIRA ticket. So we have that Slack community. You can also, propose different ideas, ask somebody if they want to work together on something. And then we have the KBase Educators Org that shares everything. We are also continuing to put together working groups. So we had a working group this summer that created all these different narratives that we have that are available to you right now. We are looking into building more. And I'm so sorry if you hear the trash cans in the background. It is trash day here on my street. But we are working together with more working groups to build out more metabolomics and transcriptomics. So any like RNA-seq analysis that you can do within KBase. And if you want to join these different groups, or have any questions for us or myself, please send us an email at engage@kbase.us. Submit a request to join the KBase Educators Org. Ask if you can be added to the Slack. Let us know, what course you're teaching, the topic and the grade level, when you'll be teaching it, how we can help, and if you're interested in connecting with peers. As well, if you want to access any of these resources that I've mentioned today, we do have a page, which is our blog, that serves as a landing page for all these resources, and definitely the overview. There's a lot of different hyperlinks in here that will take you to different pages, including our documentation page that has any help that you might, with any questions that you might have. We discuss all our different contributors from the summer. So please check that out. It also says how to, join the KBase org here. And you can find this as well as any other webinars on our Learn page. Under our tutorials, we actually have that multi-scale microbial dynamics modeling course page, as well as the blog for the KBase for Educators that links to all these different resources. And then if you keep scrolling down, we have the webinar. So you probably came to this page to sign up for today's webinar. And then we also have all of our old webinars linked here. Currently, they're stored in Google Drive, but we will work to also get them up on YouTube and create some YouTube playlists. And I, just as a final thought before we go to the Q&A document to answer any of your questions, all of these resources are continually being reiterated and are going to continue to grow as the community contributes to them. So I highly recommend, if you have been working with something, or you haven't been associated with the KBase community of organization, join it, share your resources, so that we can continue to build upon these different peer shared resources, and continue to improve and troubleshoot, and really make this the best that it can be for students, especially as they're, they're first dipping their toes into bioinformatics and getting this experience. And with that, I will open it up to questions and look at the Q&A document. Ben, have you seen anything in particular that looks like it hasn't?
Yeah, there was one, I think we got a lot of these, but there was one for Aaron directly that, where was it here? Yeah, talking about how students have responded to this. So yeah, maybe just curious if, I know there's some other folks from the educators group on here too, if anyone has any response to how students are engaging with it. And yeah, Aaron, go ahead.
Sure. So I put a quick response, but I can elaborate on that a little more here. So if I had to go back and do it again, I think one hurdle that the students, and this was especially true in the remote environment, I think had we been together in a classroom and I could have walked around and watched what they were doing, I think I could have sort of helped avoid some of the common pitfalls. And since this point, we have developed, the educators group has sort of developed, I believe, and please, Ellen, correct me if I'm wrong, sort of an FAQ on how to, if you get this error, what might that mean inside KBase to help the students work through this. But if I had to do it again, I would start with sort of a more general module I had the students watch one of the KBase videos, but I think actually having them in, load data, run through some sort of very simple apps, maybe even create different text boxes and things in the narrative, and really sort of outline that for them clearly. I think that would help them a lot when they come to the actual modules where they need to be running their own data. So that's one thing I would do a little bit differently. However, I think the question was really about, were the students understanding it? And through the homework assignments and through the practical exams, for the level that I was looking for them to sort of be at, it was clear that the students, the majority of them were able to work through and understand why they were doing the annotations, what those annotations meant when they put together the pangenome, why were pangenomes useful, how can we use this data? So they really were understanding. And from informal conversations that I had with the students, they were a little frustrated with with that learning curve. They wanted it to be, I think, maybe a little more intuitive, but they really appreciated working with real data and working in a system. I was able to show them some publications and things, working in a system that sort of people in the field are using on a day-to-day basis, and something that they felt that they all might be able to use again in the future. So this wasn't just sort of a one-off thing for them, and they really appreciated having sort of that real-world experience and that real-world exposure to these different modules. And I think that they really were understanding what I wanted them to, or they were getting out of it what I wanted them to. And I don't know who else is, I know Carlos was on. I don't know who else was part of the summer group. If please feel free to jump in if you want to share some of your experiences.
Thank you, Aaron. That was great. And yeah, I couldn't have said it better myself. I think the unique part about using KBase is that scientists are using it right now, and every day, and that's really how it's been developed. And as we've been learning and beta testing with all the educators this past fall, we have been creating and developing a few new resources, including, what to do when you encounter an error video, like where to go to find your questions, how to submit a JIRA ticket, which will soon be a video that's available on our KBase YouTube. And JIRA, I guess, it's a system that we use for our help desk. And so I think, along with, all of these different narratives that we have, the other tools that we have that are available are the videos that are up on our YouTube that you can go to ask questions, and really be able to figure out kind of how KBase might work more within your classroom. We have a bunch of different narrative tutorials where you can also get some inspiration, or you can talk to us. I am more than happy to talk to you about how to use KBase in your classroom, kind of like what you can do, and get you connected with other educators who have been using it, or who are interested in creating some of these new resources. I think with that, we've covered everything. I do have a question, actually. Would it be possible to demo on screen, apart from the narratives from the student perspective? Yes. Let me pull up a student module. I'll just pull up the one that Aaron showed earlier. So let me start sharing my screen again, because we do have the time to do this. Oh, whoops, I'm sorry. That's the wrong page. here we go. Okay, so if you were a student to go through one of, say, like this narrative, which was created by mostly by Stephen Biller, what you can do is, you're a student, this is what you would get. So your instructor would share this with you or share it with the org. As a student, you would make a copy. So you would say, okay, this is going to be mine. So I'll put my name in there and I will create a copy and I'll open this new narrative. So I'll open a narrative for myself. And from here, I'll see that there is some data here in the data pane. Here are all the different apps that are available. I would go through as a student to read this. I see that I have 3 other narratives that are included within this concept workflow. I go through and I'd read about Staphylococcus aureus, read more background on shotgun sequencing that's been put in here. Otherwise, you could also link things, like here is a animation that I could click on and that would take me to a video. And then data. So in this narrative, the data's already been imported, so I don't have to add the data in. Instead, I would just go down, see, okay, so I have a paired end reads that are in here. And then what you do is you say, okay, so this is the first step, are the reads any good? And I have a question to answer. So I kind of go through, and here we have the app. So this is one of the analysis that we are going to run this portion through. So it's the FastQC. And what I would need to do as a student is select the data. That's all I have to do for this app. I could look up more information about the app, about how it's a quality control application. And so those are my options right now. I would click run to send this through. And Aaron, do you have something to say?
Yeah. I just wanted, would you mind just clicking back quick on the info? So this was one area that I found really, really helpful. Is you see down at the bottom, it has a little information on how many times this particular app has been run and the time. So I had a lot of students click, and if 30 seconds went by and they didn't get a response, they were like, well, it's broken, it's not working. And so I was directing them often to this info pane. Some of these apps take a little bit of time to run, and the students need to be aware of that. And that was a great metric for kind of setting the right expectations of when they run this, how long it should take for it to finish.
Great, thank you, Aaron. Yeah, and so here we actually have the job status. So right now it was queued for 10 seconds, it's been running. I did get a warning in here, like any issues that pop up will come up. And once it finishes, I'll get the result. So I'll be able to see that. But this way I'll be able to answer the question once the app has run and I have the result. When I answered that question, and to answer it as a student, because this is now my narrative, I would double click on this and I would write my answer here. Or if you are more comfortable or more interested in using a different program to submit your question, for having your students submit their questions, you're more than welcome to just have the questions in here and have the students answer them and submit them through a different way. This is just one of the ideas that we thought of while we were putting this together to have it all in one place. And then you would move on and continue to do the other parts. And here, once you get to question 2, you would have another item in your data panel here. And so you would be able to select that object and then go through and add in all your different parameters for that application.
Ellen, there was also a question about uploading the data. Could we just quickly show them the different ways that data can be brought in?
Yeah, for sure. Thank you for looking at that, Aaron. So here we have our data panel. Right now I have one thing in here. If I click this big red plus button, what I have in here is all of my data. I have the data that has been shared with me from others. I have data that's public where I can actually search through different databases and select it. There's example data, and then I can import data. And so the way that you can import data is you can either drag and drop your data in here. You can also use Globus as the other way to import data. And there is also a way to import from other online sources like NCBI if you want to import there. And we have a bunch of videos on our YouTube on how to import your data and what to do. You know, if you have a really large file, like something that's over 20 gigabases, and how to use Globus. Zach, another one of our outreach members, has made some really great videos on how to do that. So students can actually import their own data. Like if you had class data that you wanted to use and analyze, you could easily import that. You could also import the data for your students, have the, similar to how Stephen did it here, you would have all your data already imported, and then you could have the students run all the analysis without having to import the data themselves. Let's see. And so here, now that this is finished, you can see it's finished with success. I have the result. Oh, wait, nope. That is the other one. I scanned too far forward. Okay, this is still running. But anyway, this is what an app would look like once it's finished with success. And you'd have the result and different images here. If there's any different parts of the report or depending on what analysis you are doing. Let me see. Do you have any other questions? I think a lot of them. Oh, that's a great question. So it's not always clear what parameters to use for each app. How do you educate the students on what to enter into the parameter fields for each app? Aaron, do you want to take this one?
So what I did this semester is I went through the student narrative and I set them all up to be the parameters that I wanted them to use. So really they had to sort of set their input data file, all of the parameters were set, and then they had to set up what they wanted their output file to be. So I really set all that up in the narratives that I rolled out to my students. And there were a few situations where we specifically instructed them to go in, change a parameter, and then evaluate how the data changed. If they, you know, for example, in the pangenome, what if they brought in, or a species tree, they brought in 5 species versus 10 species. What did that do to their tree? Did that change any of the alignment? So for the most part, I just set it up for them the way I wanted it to run. And if we did have them changing the parameters, it was very specific. And in many of the apps, I didn't change anything. I just used the default settings that were there, and those worked well for what we were trying to do.
Yeah, I'd like to respond to that one too. That's a key challenge, that question, and it's something my job is usually to try to teach KBase to scientists and researchers, and this question, there's not always a clear answer of that question to the professional researchers as well. What we usually say that is a good approach is we try to get people to think about, what is the strategy of like how they are using this tool inside of a larger methodological workflow. Students aren't necessarily doing that, but what we try to encourage people to do is, within each of these apps, there are documentation pages with links to publications. So, depending at the grade level that you're at, you might want to do something like, ask the students to read an accompanying publication, or like find in this publication where, this parameter is mentioned, because that's kind of the best way, even for those of us that are doing this in research career, that's kind of the skill that you kind of have to be able to adopt, is being able to like dive deep into where some of this information in the technical literature is. But yeah, it's a great question. I'll say we struggle to get professionals even to think through some of what the parameterization is. And with KBase, we try to set the default parameters of all of these things so that the tools will run, generally speaking. But that's always the case in bioinformatics, is that there are instances where what you are looking at, or the size of your data, and all of these other kind of things will change what those parameters have to be. So yeah, just looking at it as a teaching opportunity to try to encourage the students to go a little bit deeper, and sort of thinking like a scientist, like, right, I need to go and find the literature and identify some term and that kind of stuff. There's obviously challenges to that with, paid access to journals and stuff like that. But if we can ever help that, I'm happy to send any journal articles or anything, that y'all might need. So. Alicia?
There was also some great information associated with each of the apps on sort of what it's using, and the sort of a little bit of the background. And so for the students that really sort of got into using KBase, I was seeing that many of them were using, I apologize, I don't remember the name of the specific tab. It might be that info tab that talks sort of about the app and sort of the different analyses that it's running. And so there was a lot of information in there too that could help the students understand what parameters they may want to set and why they're setting them.
Alicia?
Yeah, just one other thing that I wanted to bring up, and thanks to both Ellen and Aaron for giving an awesome webinar, and our first introduction to hopefully a much bigger community that we're hoping to recruit everyone to be a part of this and to use these resources. One thing that I want to mention that had come up in a couple of conversations and on the shared Google Notes is that some of you might be working with students that have novel data. And one of the things I wanted to mention is that, we are moving towards a capability of students being able to publish a narrative. So take a narrative, make it really nice and pretty, clean it up, put on all your methods, have some nice documentation on it. And then we can actually create a static version of that that gets indexed outside of the Google login, that actually gets picked up by search engines. And we can provide a DOI for that. So for some of your students that are doing really novel research, or it might take a little bit longer for them to publish a scientific paper, we could get them a DOI product that they could then have linked to their ORCID and be able to share when they do decide to publish something. So that's a much faster sort of turnaround towards publishing some of your students' work than maybe trying to get a peer-reviewed scientific paper. So please let us know if that's of interest to you. We'd be more than happy to help you with that.
Thank you, Alicia. And thank you everyone else for being able to be here. I think that's all the questions for today. And yeah, so thank you. I'll turn it over to you, Ben.
All right. Yeah. Thanks again, everybody, for attending the webinar today. Just really quickly, I will put the link to the feedback form in the chat here. It's also in the Q&A doc, and I'll email it to all of y'all after this webinar recording finishes rendering. So thanks again, and we hope to see you all on the system. And like Ellen and Alicia said, please reach out to us at engage@kbase.us if you have any questions. Follow us on Twitter at DOE-KBase, and we hope to see you soon at one of our upcoming webinars in 2021. Happy holidays, everyone, and talk to you soon.
US/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.